Have you ever really stopped to look at the patterns in nature? I mean, the spots on a flower, the stripes on a leaf. It's not just random, is it? It's like this perfectly controlled biological art. So today, let's play detective. We're going to follow the clues and see exactly how these little biological artists create their masterpieces.
Okay, so let's start here. Take a look at these spots on this Asiatic lily. Now, if you get really close, you'll notice something kind of wild. They're not just flat little circles of color. The petal itself actually pushes up into a tiny little bump right where the color is. And that is our very first clue. This isn't just a paint job. Something structural is going on here.
So this brings up a really fascinating question, right? How in the world do plants manage to coordinate both the physical shape of a petal and the perfect placement of color? That is the big mystery we're here to solve today.
All right, like any good mystery, we need to meet the key players. In the world of plant genetics, the suspects behind all this amazing art are a group of specialized molecules. So come on, let's meet the crew.
First up, you've got the paint itself. These are molecules called anthocyans. They're the pigments that give so many flowers and leaves those brilliant reds, purples, and blues. Just think of them as the tubes of paint in nature's art supply store.
But paint doesn't just splash itself on the canvas. You need an artist. And that's where these guys come in, MYB transcription factors. These proteins are the master painters. They're basically genetic switches that decide exactly where and when to apply the color, all by flipping on the genes that make those anthocyanin pigments.
And hey, even a master painter needs a crew, right? The MYB protein, it doesn't work alone. It actually has to team up with two other types of proteins, bHLH and WDR. Together, they form what scientists call the MBW complex. This is the whole painting crew, and they are absolutely essential. If you don't have the full crew, guess what? No color gets made. Simple as that.
Okay, so we've got our cast of characters. Let's open up our first case file, the lily. The mystery here is all about those unique raised spots. How does the plant know to put the color only on those tiny little bumps?
And remember, the real head-scratcher here is that combination of shape and shade. How does the plant link the physical structure, the bump, to the color itself? Well, in a 2020 study, researchers zoomed in on two prime suspects, a pair of those master painter genes in lilies that they cleverly named MYB-19 short and MYB-19 long. Now, the key difference between these two is basically their power level. MYB-19 short, that's your standard activator. It gets the job done. But MYB-19 long, it's special. A part of its genetic code is copied 3 times, which basically gives it a turbocharge. Turning it into a super activator, it's way more powerful at switching on pigment production than its shorter cousin.
Okay, so we've got a painter and a super painter. That's cool, but it still doesn't answer our main question. What's the signal? What tells these genes to only start working after a bump has formed on the petal?
And this is the smoking gun. Researchers looked at a whole population of lilies. Some grew bumps, some didn't. And what this gel image shows so brilliantly is that the MYB-19 genes were only switched on, or expressed, in the lilies that actually developed bumps. The plants that were totally flat, they had the genes, but those genes were just sitting there, completely silent. So this reveals the whole sequence of events. And it's so cool. Step 1, the physical bumps form on the petal. Step 2, the formation of those bumps is the trigger that switches on the MYB-19 genes. And only then, step 3, does the pigment machinery fire up to color them in. And there we have it, the verdict for case number 1. For these lilies, structure dictates color. The physical shape of the petal has to come first, and that change is what sends the signal to the genetic painters telling them where to get to work. What an incredibly elegant solution. But wait, there's more. For our second case, we're shifting from flowers to leaves, and we're going to investigate a totally different kind of puzzle. These super sharp, defined markings on a plant called Medicago truncatula. It's a relative of clover.
So in a 2021 study, scientists found a completely different way of doing things. In this plant, the key suspects were 2 closely related genes, kind of like evolutionary cousins, that they named RedHeart1 and RedHeart2, or, RH1 and RH2 for short.
Now, these 2 genes, they're basically rivals. RH1 is the activator. It's the master painter that grabs onto the MBW painting crew and turns on the color. But RH2 is different. It's a competitor.
Over time, it's actually lost its ability to switch on the pigment, but, and this is the important part, it can still interact with the crew. Which, sets up a brand new puzzle. If RH2 can't actually do any painting, why is it even there? What possible purpose could a painter who can't paint actually serve?
Well, here's the brilliant twist. It's basically a genetic duel. In some cells, the activator RH1 gets to the painting crew first, binds on, and bam, you get color. But in other cells, the competitor, RH2, beats it to the punch. It binds to the crew, and since it can't actually make pigment, it just sits there, blocking RH1 from doing its job. It's what's called a competitive inhibitor. It's a saboteur.
And this constant competition is the secret to the whole pattern. This battle between the activator and the competitor, happening at the cellular level, is what creates those incredibly sharp, clean boundaries. It's like putting down painter's tape. The presence of that RH2 competitor makes sure that color only shows up in very specific, well-defined zones.
So, after investigating these two cases, what's the final verdict? Well, we've uncovered two totally different strategies that nature uses to get to the exact same goal, creating a beautiful, precise pattern.
Let's just break it down side by side. In the lily, right, the mechanism is structure-led activation. It's all driven by that powerful superactivator gene that only kicks in after a physical bump forms. But in the Medicago plant, the whole game is competitive inhibition, that duel between an activator and a competitor gene that creates those sharp edges. Two different tools from the toolbox, two totally stunning results. And that's really the crucial takeaway here. Nature doesn't have just one way to paint a masterpiece. Its genetic toolbox is just packed with all these elegant, diverse, and creative solutions to complex problems.
It really makes you look at every leaf and every flower a little bit differently, doesn't it? And it leaves us with one last really exciting question. What other amazing genetic stories are just waiting out there for us to discover in the beautiful patterns that are all around us?